Hi, my name is Brent Roger. I am a biology major with a concentration in pre-med, and I am doing my thesis on the presence of antibiotic-resistant bacteria in recreational fishes of coastal Louisiana. First, I would like to start off with a brief background on the seafood industry in Louisiana. According to Louisiana Wildlife and Fisheries, 49,590 registered commercial fishing licenses were issued in 2019. In 2008, there was a survey in Louisiana that had numbered over 700,000 anglers in the state alone. 1.2 billion pounds of fish were landed in Louisiana in 2012, and 1.6 billion pounds of fish were landed in 2016. With increases in the demand for seafood, there are also increases in the likelihood of these fishermen and fish processing industries coming into contact with antibiotic-resistant bacteria. Now, as you can see here, in 2019, the CDC reported over 2 million people affected by antibiotic-resistant bacteria, and around 33,000 people died from antibiotic-resistant bacteria in the United States. Now, how do these antibiotics end up in the environment to lead to antibiotic resistance? Many studies have confirmed antibiotic resistance in varying water and sediment sources in Louisiana. A study from Hollis and Ahmed in 2013 showed that 80% of antibiotic sales went to livestock and aquaculture. Now, these antibiotics that the farmers and agriculturists give the animals and fish in the fish farms, they end up in the waste and water, and they get drained right into the environment. Also, another study shows 266 million courses of antibiotics were prescribed to outpatients in 2014. The CDC reports that 30% of these outpatients improperly dispose of the antibiotics they use, which means they just either toss them down the drain or they throw them out into the environment, and this is very harmful. Research done in Dr. Bhupathi's lab has confirmed the presence of antibiotic-resistant bacteria and antibiotic-resistant genes in sewage treatment processes here in Thibodeau. Now, other ways antibiotics end up in our environment are pharmaceutical waste, pest control, and even our cleaning products that end up down the sewers and into our bayous and rivers. How do these antibiotic-resistant genes spread among the bacteria? Now, some are naturally resistant to certain antibiotics. The bacteria either have a thick cell wall that prevents the antibiotic from attacking the bacteria, or some other bacteria have internal mechanisms which pump out the antibiotics as they come in, so that sort of neutralizes the effects of the antibiotics from working. Other ways are due to selective pressures from antibiotics in the environment that cause the selection of the antibiotic-resistant bacteria to flourish and pass on their genes, which leads to horizontal gene transfer from the bacteria. Now to discuss vertical and horizontal gene transfer. Vertical gene transfer is more of a random beneficial mutation from asexual reproduction of the bacteria. This is when the bacteria over multiple times divides on its own, and it has a chance to mutate. Now, this mutation could either not have any effect at all, or it can be beneficial, and on that rare occasion, that mutation could be antibiotic resistance. Horizontal gene transfer is completely different. It has three different modes of transferring, possible transferring of antibiotic-resistant genes. That is conjugation, transduction, and transformation. Conjugation is sexual reproduction of bacteria, where they exchange cellular DNA through the pilus. Now, they copy their DNA, and whenever they exchange their DNA for sexual reproduction, they can transfer that extra copy of DNA to the other bacteria. The second mode is transduction. Now, this is where a bacteriophage transfers antibiotic-resistant plasmids from one bacteria to the next. It's kind of like a free ride. They inject their DNA into the bacteria, they multiply, they can pick up those floating antibiotic-resistant plasmids, and it can get mixed in with the virus DNA. And when the other viruses move on to a different bacteria and inject their DNA in, they can inject those antibiotic-resistant plasmids to the other bacteria. Now, the last mode is transformation, where the donor bacteria gives the recipient bacteria the antibiotic-resistant plasmid. And this recipient bacteria sort of phagocytizes the DNA. It takes in that free DNA, and that DNA becomes incorporated in that bacteria, and that bacteria gets the antibiotic resistance. For my experiment, I wanted to identify antibiotic-resistant bacteria in seafood samples and confirm their antibiotic resistances. There have been no reports of antibiotic resistance from seafood in this region, and this should prove that there is a presence here. Now, there have been other studies elsewhere where they test antibiotic-resistant bacteria in water sources and fish farms. There has not been any conclusive studies done on the seafood here in our region of Louisiana. My fish samples were taken from caught fish from the fish market. I want to make a note that these samples are from the John Fulse Culinary Institute. So there are possible bacteria from human handling as they were caught from the ocean and brought to the Institute. I did my study from redfish, black drum, and Atlantic croaker, all of which were fished right off the coast of Louisiana. And as you can see from this this map, this is the population of redfish. I looked up different populations of the black drum and Atlantic croaker, and they all overlap along the coast of Louisiana. Now, my methods to identify the bacteria are as follows. I first collected and grew my samples in tryptic soy broth, or TSB for short. I then isolated distinct colonies on tryptic soy agar, or TSA for short, plates, and followed the same process over and over to get a pure cultured colony on my plates. Then I used the biolog method to identify the bacterial colonies from each fish. Lastly, I used the Kirby-Bauer test to show antibiotic resistance in the bacteria from my samples. And this test will show that there is an antibiotic-resistant presence here in Louisiana. Now, to identify my bacteria from my samples, I used the biolog. This is a device that uses different substrates and carbon sources to identify the bacteria. And the way it works is you take your bacteria and you place them in a 96 well plate, as you can see in the picture below, and you incubate that plate for about a day or two. And what happens is the bacteria sort of uses these sources for food, and the machine detects these changes in the metabolic rates. And by using these changes in the rates, it can pretty accurately identify the bacteria. The biolog machine identified several different kinds of bacteria in my samples. Listed below are the big ones that showed the most antibiotic resistance. These include Pseudomonas aeruginosa with a 90.7% match on the biolog machine, Pseudomonas mendocsana at an 85.2% match, Morganella morgani with a 98.6% match, Enterococcus sulphureus 63.1% match, and Bacillus stephanensis with a 71.9% match. Now, the two at the bottom are lower, but this is in the normal range. The normal range in the biolog machine is 60% or higher. Now, each of these bacteria are unique in its own right. Pseudomonas aeruginosa is a big one. It's a gram-negative opportunistic pathogen, commonly known to cause pneumonia or sepsis in patients post-operation or catheter. It can be found in man-made environments like in the workplace, especially in a hospital environment. It can also be found in soil and water, so pretty much anywhere. Pseudomonas mendocsana is a gram-negative opportunistic pathogen, but it's only rare in that case. Usually it's nosocomial infections, usually post-operation. Morganella morgani is a gram-negative rod found in the environment, and it has been found in trace amounts in the intestines of humans and animals. And usually it's only seen as an opportunistic pathogen or nosocomial pathogen in like infections like from the hospital post-operation, like most bacteria like E. coli. If it gets into an open wound, it's very bad for us, but if it's in the proper place like the intestines, it can actually do us some good. Enterococcus sulphureus is a gram-positive cocci known to cause urinary tract infections, and it can be found in various different environments. Bacillus stephanensis is a gram-positive rod. Now, not a lot of studies have been done on this bacteria, but there has been a case that this bacteria has caused food poisoning. The Kirby-Bauer test was the next step for my experiment to confirm the antibiotic resistances of my bacterial samples. Now, here I use tetracycline, sulfonamide, bacitracin, penicillin, ampicillin, vancomycin, clindamycin, and streptomycin for my plates, but I also use cefoperazone, ceftazidime, impenem, and piperacillin for Pseudomonas, as you'll see in upcoming slides. These are second and third generation antibiotics, unlike the others, which are first generation, and are much harsher. Each of these antibiotics are used to target the protein production of the bacteria, like the first two, tetracycline and sulfonamide. While the others, antibiotics, they attack the cell walls and membranes of the bacteria instead. This test measures the zones of inhibition for each antibiotic on the plates with the bacterial isolates. I made my measurements using a caliper, and using the next couple of slides ahead, I was able to make a chart on if the bacteria were resistant or susceptible to the antibiotics. And as you can see for Pseudomonas aeruginosa on the right side, it was completely resistant to the first generation antibiotics. So I went for some advice and used some of these harsher antibiotics to see how resistant this bacteria really is. Now, these two charts and these two slides are examples. The full list that was given to me was in the lab as I made my measurements. As an example is Pseudomonas aeruginosa. As you can see from the plates before, Pseudomonas aeruginosa had complete resistance to the first generation antibiotics. There was no zones at all. However, on the second plate with the second and third generations, I got zones around the antibiotics. For impenem, cefoperazone, and piperacillin, I got intermediate resistance. The zones were about either 12.3 millimeters in diameter or 15 millimeters in diameter. And from this chart, I looked, and the I here means intermediate resistance. So it had intermediate resistance for that antibiotic. However, ceftazidime was the most effective for Pseudomonas aeruginosa because the diameter was 23 millimeters. And on this chart, if you see either the ceftazidime, you can find the label here, 23 millimeters, that is within the S range, so susceptible. Again, this is just an example of the chart that was given to me. It's a huge list. And these have some of the first generation antibiotics. A lot of the bacteria are about the same when it comes to the list. I made sure to double check on all my bacteria whenever I made my chart coming up. But as you can see here, if it's anywhere above 15 to 18 millimeters, the bacteria is susceptible to the antibiotic. But if it's around the 15 to 14 or even 12 to 15 range, it's intermediate. And if it's less than 12 or even no zones at all, then it's completely resistant to that antibiotic. I made this table to sort of simplify all the data and information from the charts before and from the chart in my lab to sort of give you an idea of how resistant these bacteria are. Pseudomonas mendocina, it was resistant to all the antibiotics. For tetracycline, I got a 3.6 millimeter zone, so that was within the resistant range. I got 10.6 millimeter for sulfonamide for Pseudomonas mendocina, and that was in the resistant range as well. Clindamycin was 10 millimeter, so that's resistant, and vancomycin was 6 millimeter, which is also resistant. Now, Pseudomonas aeruginosa, as I said before, it was resistant to all the antibiotics. It had no zones at all. Had to use the harsher antibiotics to actually get results. This could be because Pseudomonas aeruginosa made that green biofilm. If you go back and see those plates, it's turned green. That's because these bacteria produce a biofilm to keep a protective layer and sort of keep those antibiotics from reaching the cell walls and membranes of the bacteria. Morganella morgani showed quite a few resistances as well. Sulfonamide had a 15 millimeter diameter zone, so it was intermediate. Streptomycin had 9.5 millimeters, so that was resistant. And ampicillin was 11.3 millimeter, and that was resistant as well from the charts. Bacillus bihenstephanensis had quite a few resistances as well. It had 9 millimeter tetracycline, which was resistant. Clindamycin was 14 millimeters. Now, my duplicates for this, it bounced between 14 millimeter and 15 millimeter. So that made it kind of difficult to decide between what to put it as. I put it resistance because 14 millimeters is within that resistant range. The 15 millimeters makes it intermediate resistant, so I made a note there on my table that it's between those two. And then there's, of course, Enterococcus sulfuris. This bacteria gave me some troubles in my lab experiment. I made multiple duplicates with Enterococcus sulphureus. One of my plates, I actually got it to grow, and as you can see, I got like it showed resistance to tetracycline, sulfonamide, bacitracin, and penicillin, but it had a zone for ampicillin, and it showed intermediate resistance. The other plates, it had literally no growth on my plates. This may have been because of lack of nutrition, or maybe I just didn't have it in the right environment to grow bacteria. So the results for this is really like inconclusive for this bacteria. I would have to redo my experiment for Enterococcus sulphureus to sort of confirm these results for Enterococcus. Now, there are other experiments that can be done with my bacteria samples. One experiment could be the molecular assay, which would look for the antibiotic-resistant genes of my bacteria. Another would be an experiment on the concentrations of antibiotics and how the bacteria grows in these different concentrations. It's called the concentration assay. Another is EC50, which would look at the concentration of antibiotics needed to kill 50% of the bacteria. I wanted to do an example of a concentration assay. So I managed to complete a duplicate of Bacillus bihenstephanensis and sulfonamide. This chart shows the concentration of sulfonamide used and how the bacteria grew over a span of 2 weeks. I used a spectrometer at 600 units to measure the growth of the bacteria each day. And interestingly, this chart shows that the bacteria were able to thrive even under the heavy concentrations of the antibiotic. Of course, further research is needed to prove this statement, but the results were interesting to see from this duplicate of the bacteria under the different concentrations of the antibiotic. I wanted to give a special thanks to my major professor, Dr. Raj Bhupathi, for his help and for allowing me to use his lab and materials for my experiment. I also wanted to thank Ethan Naka for his guidance. And I want to say thank you to Mr. Jean Daig for providing samples from his seafood class at the Culinary Institute. And thank Dr. Corbin for providing stronger antibiotic suggestions for my Pseudomonas aeruginosa. These are the articles I used for my PowerPoint and research. And please ask questions or make comments in the comment section under this video. And thank you for watching this video.